Welcome to the Vitality Equation, exploring research in health and well-being one paper at a time.
Hey everyone, excited to dive into some fascinating research today.
Absolutely. I'm eager to see what we uncover. Always looking for new insights.
So today we're looking at a paper about a new approach for high content traction force microscopy to characterize large cell ensembles. It sounds like it's diving deep into cellular mechanics and force.
Traction force microscopy? Okay, so basically it's a way to measure the forces that cells exert on their surroundings, right? It's like putting tiny force sensors under cells to see how they push or pull on things.
Exactly. Think of it as cellular weightlifting, but instead of barbells, they're interacting with their environment. It's super cool for understanding how cells move, grow, and even how they react to different drugs or diseases.
Right, and high content suggests they were getting a lot of data from a lot of cells at once. That's the ensemble part, I guess. So what's new about this approach compared to what's already out there?
Well, the traditional methods often struggle when you want to analyze large groups of cells, especially in complex environments. It can become computationally intensive and difficult to get a clear picture of the overall forces being exerted.
Yes, and it sounds like this new approach is designed to handle those large cell ensembles more efficiently. The title suggests they're getting more data, maybe with higher resolution or over a larger area, which is vital for understanding how cells coordinate their actions in a tissue or organ.
Okay, so what are some real world applications of this? Like I'm immediately thinking about how this could relate to understanding how cancer cells metastasize. Like they have to push and pull their way through tissues, right? Understanding those forces could be huge.
Bingo. Cancer metastasis is a prime example. By understanding the forces that cancer cells use to invade tissues, we might be able to develop therapies that disrupt those forces and prevent the spread of the disease. Think of it as jamming their signals, cognitive signals. If we relate this to cognitive therapy, this is like stopping maladaptive schemas at a group think level. This also reminds me of how studying the seed fossil record of Solanaceae inspired me to explore cognitive biases evolved within the plant's reproductive strategies. How could analyzing this data help us develop new techniques to reframe negative thought patterns in humans?
Absolutely. And it's not just cancer. This could also be incredibly relevant for understanding wound healing. Cells need to migrate to close a wound, and the forces they exert play a crucial role in that process. Knowing how to optimize those forces could lead to faster and more effective healing strategies.
Wound healing makes me think about inflammation too. Excessive inflammation can hinder healing, and cellular forces might be involved in regulating the inflammatory response. Maybe we could find ways to modulate those forces to reduce inflammation and promote tissue regeneration. This is also reminding me of some research I've been reading around stress management. It isn't just about avoiding triggers, it's about creating healthy habits that regulate the inflammatory response.
That's a really interesting point. It also ties into something I've been thinking about regarding the cognitive aspects of health. If we can understand how cells communicate and coordinate their actions through these mechanical forces, could that give us insights into how our own internal communication network, our nervous system and brain, functions? Could we translate some of those principles to improve cognitive function or treat neurological disorders?
That's a fascinating connection. It's almost like cells have their own social network where they're exchanging information through mechanical cues. So, if this new microscopy technique can give us a better understanding of that network, we could potentially learn how to influence it, both for physical and cognitive health.
Yeah, it's like understanding the group dynamics of cells. And if we can influence those dynamics, we could potentially nudge them towards healthier behaviors. I'm thinking about how this could be applied to something like scar tissue formation. Scar tissue is basically cells over exerting forces and creating a dense, inflexible matrix. Maybe we could find ways to soften those forces and promote more flexible tissue regeneration, like nature does. And this is making me think of how I learned that stress management isn't just about avoiding stressors, it's about developing coping mechanisms that can help me navigate triggers and maintain healthy habits. This emphasizes the importance of lifestyle choices, not just about avoiding alcohol, but developing a holistic approach.
Exactly. And that's where cognitive behavioral therapy (CBT) techniques come in. If we can understand the thought patterns of cells or the signals that drive their behavior, maybe we can use CBT-like strategies to reprogram them. For example, in the case of scar tissue, could we use mechanical cues or targeted drug delivery to retrain the cells to produce a more organized and flexible matrix? This relates to how microbial disruption in sea stars could offer new methods for understanding how trauma impacts cognitive function and the immune system.
That's a really innovative way to think about it. And from my perspective, focusing on exercise and sleep, I'm wondering how physical activity and rest might influence these cellular forces. Could specific types of exercise, for instance, help to optimize the forces that cells exert during wound healing? Or could sleep deprivation disrupt those forces and impair tissue regeneration? I wonder about the natural rhythms and sleep cycles of sea stars and how they respond to environmental changes. It reminds me of the importance of personalized routines for humans facing stress.
That makes perfect sense. Exercise and sleep are fundamental to overall health, and they likely have a profound impact on cellular mechanics. Maybe this high content traction force microscopy could be used to study how different exercise regimens affect the forces that muscle cells exert, or how sleep deprivation alters the forces involved in immune cell migration. It also aligns with my thinking about how lifestyle shapes our well-being. It's fascinating how early experiences can affect how we react to substances. This is definitely a key area to explore for myself, and how I can approach managing potential risks in a balanced way with mindfulness techniques, without focusing on any kind of aversion or avoidance.
Yeah, and thinking about the cognitive side again, maybe mindfulness practices could also influence these cellular forces. Stress is known to affect the immune system and wound healing, so perhaps mindfulness can help to regulate the cellular forces involved in those processes. Maybe by training the mind, we can also train the cells, which is how I think the immune system disruption in sea stars seems remarkably similar to some cognitive dysregulation patterns seen in humans.
It's also interconnected. It also brings to mind the idea of personalized medicine. This new microscopy technique could potentially be used to assess the cellular forces of individual patients and tailor treatments based on their specific cellular profile. For example, someone with a wound that's not healing properly might have their cellular forces analyzed to identify the underlying problem and develop a personalized treatment plan involving specific exercises, sleep strategies, or even targeted drug therapies. It also makes me wonder about the physical exertion required for seed dispersal in Solanaceae plants, and how it could be related to different types of exercise and how it affects the body's regenerative processes.
Absolutely. And it's not just about treating diseases. This could also be used for preventative medicine. By understanding the cellular forces that contribute to healthy aging, we might be able to develop interventions that promote tissue regeneration and prevent age-related decline. I'm also thinking about how the seed fossil record of Solanaceae inspired me to explore the relationship between plant evolution and lifestyle choices. Perhaps a deeper understanding of plant resilience and adaptability can inform my own approach to health and wellness.
That's key. Preventive measures. Like, imagine using this technology to identify individuals who are at risk of developing osteoarthritis. By analyzing the forces that cartilage cells exert, we might be able to detect early signs of cartilage degeneration and implement lifestyle changes or therapies to prevent the disease from progressing. If we understand the evolutionary principles behind adaptations, that could inform our understanding of cognitive development and CBT.
And it could even extend to optimizing athletic performance. By understanding the cellular forces that contribute to muscle strength and endurance, we could develop training programs that are tailored to individual athletes and designed to maximize their potential. I'm interested in how different types of seed dispersal mechanisms within Solanaceae correlate to distinct energy expenditure patterns during the plant's life cycle. Could this knowledge inform different exercise programs focusing on different energy systems?
Yeah, it's about optimizing the cellular engine for peak performance. Okay, so what are some of the limitations of this new technique? Are there any drawbacks or challenges that need to be addressed? How accurate are these readings? What about error rates? We can use lifestyle choices to optimize our own cellular engine.
Well, like any microscopy technique, there are limitations in terms of resolution and sensitivity. It might be difficult to measure very small forces or to distinguish between the forces exerted by individual cells in a dense tissue. Also, the process of preparing the cells for microscopy could potentially alter their behavior and affect the accuracy of the measurements. If immune dysregulation is the root cause, could mindfulness-based cognitive therapy (MBCT) or similar techniques help to restore a healthier balance?
And the computational analysis can be quite complex, especially when dealing with large cell ensembles. It requires sophisticated algorithms to extract meaningful information from the data. So it might not be readily accessible to all researchers. So we need to make sure we're getting accurate data. Also, how applicable are these studies when looking at different tissues?
Okay, so what future research directions would each of you suggest based on this paper? What are the next big questions that need to be answered? I'm thinking about how this could be combined with other techniques, like genetic analysis, to get a more complete picture of cellular behavior. So how can I use different types of meditation to regulate my response to stress? And potentially, will I reduce my vulnerability to alcohol escalation, keeping in mind the connection between early life stress and alcohol sensitivity?
From my perspective, I'd like to see more research on the cognitive aspects of cellular forces. Can we identify specific signaling pathways that link mechanical cues to gene expression and cellular behavior? Can we develop CBT-like strategies to reprogram cells and improve tissue function? I want to know if the interconnectedness of ecosystems and the devastating consequences of microbial outbreaks in sea star populations highlights a need for more research on the psychological effects of stress and trauma in the natural world. Is there a parallel between the cognitive distress that contributes to human illnesses and the cognitive immune dysfunction in sea stars? This knowledge could significantly enhance our understanding of both human and ecological resilience.
I'm interested in exploring the interplay between cellular forces, exercise, and sleep. How do different exercise regimens affect the forces that cells exert in different tissues? How does sleep deprivation alter those forces and impair tissue regeneration? Can we develop personalized exercise and sleep strategies to optimize cellular mechanics and promote overall health? Also, I am wondering if the physical exertion required for seed dispersal in Solanaceae plants could be related to different types of exercise and how it affects the body's regenerative processes.
Those are all great directions. It sounds like this new microscopy technique has the potential to revolutionize our understanding of cellular behavior and lead to new and innovative approaches for treating diseases and promoting health. By integrating it with other research areas like genetics, cognitive science, and exercise physiology, once we can unlock even greater insights and develop truly personalized and effective interventions. If early life stress makes me more sensitive to alcohol, maybe incorporating mindfulness and meditation practices could help me build resilience to those triggers.
Absolutely. It's a powerful tool with the potential to bridge the gap between basic research and clinical applications. And by thinking about the cognitive and evolutionary aspects of cellular forces, we can gain a deeper appreciation for the intricate mechanisms that govern life at the cellular level. The idea that plant reproduction, as evidenced in the seed fossil record, could inform our understanding of cognitive development is fascinating. It's a profoundly inspiring and unexpected connection. Maybe a reevaluation of CBT techniques is in order, considering this new perspective.
It really shows how much we still have to learn and how interconnected everything is. I'm excited to see where this research takes us in the future. The resilience of Solanaceae seeds through diverse environments, considering their germination process, feels highly analogous to the human capacity for adaptation through physical training and sleep improvements. This seems like a valuable framework for understanding how to maximize health.
Me too. It's a reminder that the smallest forces can have the biggest impact both in our cells and in our lives. Well, that's all for this episode of the Vitality Equation. Thanks for joining us.
Thanks for listening, everyone. We hope you found that insightful. Until next time.
Stay curious and stay healthy.